let me tell you something about what you're in for. Lewis told me this was to be a public lecture, and I took the word public seriously, so I have attempted to make a lecture with no formulas. I didn't quite succeed, but I think I came close, even though I'm going to talk about a mathematical concept during part of this. Here we go. So an outline of what I'm going to talk about is the history of the human race's acquisition of genetic sequences, at least up to this time. I'll talk about a computational problem, really, of finding shortest paths between 2 points, and connect that with some of the work that was critical in analyzing and obtaining all these sequences, and then move into a few slides about the diversity and commonality of the genetic material all of us in this room carry around. And I'd like to end with saying a few things about race, even though that is not a subject that I'm professionally expert in. I, like many of us, am fascinated by that. So that's where I'm going to end up. So Lewis talked about 1974 being early in this subject, but someone who was earlier in this subject is this extraordinary woman who's passed away now, but her name is Margaret Dayhoff. And in the 1960s, when protein sequences were being obtained, but nucleic acids were extremely hard to get your hands on, she published, pioneered this collection of books. And I'd like to tell you a little bit about her and about these books. Here's a brief summary of something called the Atlas of Protein Sequence and Structure. And I hope it's in the library somewhere in Singapore. These are worth looking at. They're everything that was known at that time. So in this 1965 version, there were 93 basically mimeograph pages. But by 1966, with Richard Eck, she published this 215-page volume, which contained all the sequence information that was known at that time, plus essays about how to analyze them and so on. Dayhoff matrices, if anyone's in bioinformatics, Dayhoff matrices, this is Margaret Dayhoff. She was from physics, and she did a Markov chain analysis that was a mystery to many of us, including Terry Speed and I, in exactly what was going on. Eventually, I think Martin Vingren figured that out some decades later. But one thing I want to point out is that Richard Eck, whom I don't know a lot about, got himself made first author of this second. Let's see, there's not a. Anyway, second author, but by the third version in '67, she had got herself in first place, and in all subsequent versions, there were no men as co-authors of the book. It was only women. The men, I think, took care of the coffee, and maybe did some really routine programming, but they were not in the action there at all. But so to put it in perspective, how much $3.50 was in 1966, I had an NSF fellowship, and I supported myself and a wife, and the next year a daughter, on $300 a month. So this is about 1% of a graduate student's salary. Now, 1% probably would seem like a lot of your salary, would seem like a lot today, but imagine you get in printed form everything that was known about genetic information. It would be a heck of a bargain, wouldn't it? So there's one more picture of Margaret and her crew. And in this one, Margaret's on, excellent. So Margaret's on the right. So this is Margaret here. Notice the cool computers there. I don't know, there was a woman named Lois something, and perhaps that's her. The men you see are standing just behind. About the next year, they weren't even in the room. And if you can see, that's Mona Lisa on the wall back there. She was a tough customer, I must say, but I liked her. And she produced these curated volumes, and she wanted to understand the sequences before she put them out. And the advent of modern sequencing really sort of put her way of doing things out of business. She would be again in business today. So in the early, she died maybe in the mid-'80s, but in the early '80s, when the nucleic acid information was being collected, she just didn't have the mentality for just collecting it all in and putting it out. So the history of nucleic acid sequencing, 1953, the double helix, 1963, there's a paper that published about a 70 base pair, or 70 base RNA sequence, and that was probably 20 or 30 human years of work to produce that. It was extremely hard to sequence nucleic acids. By 1975/1976, the fast modern methods were invented, and then suddenly these things started coming out, and governments both in Europe and the US realized they had to collect them. And I presented slides that looked like this for years. It looked just like this in 1984. It looked like this in 1988. Even looks like nothing was going on then. Of course, this is exponential increase. Lots was going on. So, of course, what you need to do is take the logarithm, naturally. So, in Europe, I think they got going a little more in 1982. The people at Los Alamos, which where this was formed, had trouble getting the sequence, getting very many of the sequences in. So it's a very sort of slow ramp up. And then it kind of had this exponential increase. And I've stopped at 2001, but sharp eyes will notice there's a bump up there. So in 1995, the Human Genome Project was started, and there was this orderly procedure to map and sequence the human genome. And then in the late 90s, this guy named Craig Venter decided, I'm going to start a private company and just sequence it myself, forget these guys who don't need the public project. And what you see in that bump right there is the adrenaline that Craig Venter put into the genome projects. And there was this excess of people desperately trying to get the sequence done and out. And I think we probably owe Craig Venter having the genome project finished 5 years early, or the first good draft 5 years early. So anyway, so there's two features of this that you can notice. But the doubling time through this slide, it may be a little more now, was about 1.5 years. Here's the sort of numerics of this, and part of the reason I've lost my died on me at any rate. The millionth base came somewhere toward the end of 1982, and the people at Los Alamos had a champagne party to celebrate putting in base number 1 million. 2 weeks later, they put in base 2 million. And that's kind of the way this thing went. They couldn't keep up for years and years and years with the rate at which the sequences came in. And one of the things I love about that is all of us who publish things, who write books, we love books. And so my friends at Los Alamos, of course, after 1984, they had 3 million base pairs. They published a book. Absolutely useless. I wish I had bought these because they're kind of neat. There it is, but they were not in any usable form, of course. And it was a 2-volume set. And 1 year later, they published, of course, a 4-volume set with 4 million base pairs. And 1 year after that, an 8-volume set with 8 million base pairs. And even then, they finally gave up. But I wish I owned these books just for sort of sentimental value. But once again, you couldn't imagine having all the genetic material printed in a book now. It would be just as useful as those were, though. So what's going on with the sequencing around 1995? The Human Genome Project was first talked about in '85, officially began in 1990. And by 1995, it was this amazing explosion. And this 95 publication of the complete genome sequence of this bacterium, Haemophilus influenzae, about 2 million base pairs. And really the amazing thing about it, I think, was the computational method they used to put it together. I was surprised that they could accomplish that. And I should say, because computers is my theme, that computational science and modern molecular biology kind of grew up together. It's obvious we would have computers without the molecular biology motivation. But modern molecular biology simply could not be without computers to gather the information, without computers to store the information, without computers to access the information. It would be impossible. We'd be in a completely different phase. So even for non-computational biologists, this is really a key thing. The year later, the yeast was sequenced, which is an order of magnitude larger, 12 million base pairs. In 1998, the worm C. elegans was published, 100 million base pairs. You kind of notice the clicks in 10. And Celera Genomics, Craig Venter's company, to practice for sequencing the human genome, they, with a public effort at Berkeley, together did a sequencing project of the Drosophila, this fly, of 140 million base pairs. And then 2000 was the first announcement of the complete sequence of the human genome. It'll never be completely complete, but an amazing progress in a very short number of years. So today, there are 485 complete genomes. Of these, about 461 are prokaryotes, 24 eukaryotes, like eukaryotes are things with nucleus like you and I. Of those, 2 mammals, man and mouse. In addition, there are about 400 draft genomes. That isn't a finished product, but a lot of it's out there. 508 genomes in progress in addition to that. And a year ago, there were 130 billion bases in the NCBI database. Put that in perspective, we're about 3 billion base pairs in our own genome. So now I want to talk a little about computer analysis of these sequences. And there are a whole bunch of problems, some of which are to take the sequences and try to study the ancestral relations. And that's how I got started in this. The problem that we worked on that first summer at Los Alamos, we had a huge amount of data. We had 27 cytochrome C sequences. And it was a huge, you've got to do all pairwise comparisons. It was a lot of, and our goal, these are proteins that help with electron transport in the cell, and they preserved over a huge amount of evolutionary time, and we were trying to estimate, to infer what the evolutionary relation to the family tree was amongst these protein sequences. And that problem is still of interest today, not just for cytochrome C sequences, but for genomic sequences. So finding evolutionary relationships is a big computational problem. There are lots of subtle patterns in sequences, and one of the favorite ones these days is trying to find where proteins bind to the DNA. Those patterns are not exact, and they're very, very fuzzy and hard to locate. So it's another big computational problem. The problem that Temple Smith and I got our name joined together, where he became the first name and I became the last name. He told me once he was introduced as Waterman, and he kept going right on and gave the talk, which I think was pretty funny, is to find, so this really came about when in 1978 it was discovered that in organisms like us, unlike in bacteria, there are introns amongst the coding region for a protein. So there may be 600 letters encoding a protein, but it may in our genome be spread out over 200,000 letters of genomic sequence. And so it's more than just us, but immediately realized that these comparisons, which were to compare all of one sequence to all of another, were not relevant anymore. You needed to find pieces that looked a lot like one another. And that was our motive. And then, of course, the assembled DNA sequence from short randomly located reads are the genome projects of sequencing the genome. Once you have that, if you have this picture of the 600 letters spread out over 200,000, you've got to figure out where the genes are, where the coding regions are amongst this huge amount of genetic material. Our genomes have about 2% genes. So it's again, a huge computational problem. The non-formula formulas that I'm trying to sell you today is a problem of route finding, and most of the things I just described can be fit into one of these problems. And so, for the point, although Singapore is not a huge place, there are lots of roads. So there is a challenge to find your way from one place to another. At least it is for an outsider. So at any rate, the problem I want you in your head is that you've got a complicated road map, and you're at city A and you're trying to get to city B with the minimum number of miles. And when I originally put these maps together that I'll show you in a minute, I was flying from San Francisco to Toronto for this Gairdner Award, and so I used Toronto as the destination. And it sounds easy, but when you start looking at maps, it suddenly becomes harder to think of how you'd make this analytical or how you'd really solve this problem. And this is from a road map from, I think, the 30s. It was before. It was certainly before World War II. And this was the time when America was starting to. This is New Mexico, incidentally, and America was thinking about how to make roads that went across the country, and they originally wanted these roads with that were multiples of 10. And so Route 60, which goes basically, and it's now called Route 66, and you may know the song, Get Your Kicks on Route 66. But originally it was Route 60, which wouldn't rhyme with that. And the fuss was it went from Chicago to Los Angeles, but it wasn't transcontinental, so it couldn't be 60. So they changed it to 66. At any rate, in this map, it looks like a simple map, and it really is. Does anyone happen to have one of these? I'd love to point it. I guess this is working for a moment. So this is an Indian reservation. If you're going to go to New Mexico and buy Navajo rugs, you would go to the auction at Crown Point if you knew what you were doing. Otherwise, you'd go to Santa Fe and pay more money. So here's Route 60, and down here is an Indian pueblo at Zuni, lava beds, simple. Come to Albuquerque, and there are two ways to go to Santa Fe from Albuquerque. One is right up what's now the freeway. The other one is called the back way. You go around here. The Golden Inn is a famous bar. It's in songs. Madrid is full of hippies, people still doing weaving and that sort of thing. So which of those would be shorter is not obvious from looking here. Lamy, and of course, Los Alamos isn't on this map yet because World War II hadn't happened yet. But when it did, the scientists got off at Lamy, which was the train stop, and went into to Santa Fe to get a ride up to Los Alamos, which was the secret place where the bomb was made. So even here, there's some complications on how you'd choose your way. A little further, and remember, and this is a lot, we're at Chicago, and it's a mess. And how you'd figure out getting your way through here in the minimum number of miles is not at all obvious. Kind of the thing, you have these junctures where there are several ways to have gotten there and leave, and what a mess. And at Chicago, you're not to Toronto yet anyway. So The neat way to think about this is something called dynamic programming, which somebody once said is neither dynamic nor programming. It's a little bit programming, but it's definitely not dynamic. It's very static. And all of our instincts when we're thinking of how to get our route from San Francisco to Toronto, what we'll ask ourselves is, where do we go next, right? That's not the right way to solve the problem. The way to solve the problem is to think, say, if this city were on the best way, how would I have driven into this city? So you think backwards. Where did I come from instead of where I'm going? And that was Richard Bellman. Many people did this. Richard Bellman invented this name, dynamic programming, which became very famous. And so the idea basically is, imagine we're at city C and we want to ask how we got there. And there are only three cities adjacent to C. So we can only come into city C from B1, B2, or B3. And we're going to imagine we solved the problem from San Francisco to B1. There may be a terrible tangle of roads, but we know the answer of the shortest way to go from one of these places to the other. And so this is the only formula. The mathematicians are disappointed, the biologists are happy. At any rate, so the symbolism is the shortest distance is just distance from San Francisco to C. So the distance from San Francisco to C has to be the best one of going from San Francisco to B1 and then B1 to C. Now this is, you can look this up on the map. This one we've already computed, we think. So, or the same thing for city B2, B2 to C, whatever that mileage, which again is on my map, plus this other part, which we've originally solved. So you just take these three sums, pick the smallest, and you're done. And so the simple version of the idea is that you recursively compute that. Now, when you leave San Francisco, there are only three places you can go. That's easy enough, so you've got these guys. Then at the next level, you get to these, to this level of city from the ones before. So this one is the minimum of those three numbers, those three, those two, and that's a simple calculation. And you just keep doing that. So by the time you get out here, there may be an exponential number of ways of going from San Francisco to there, but you've got all the numbers built up at that point. So it's recursively, and finally you get the distance to Toronto. And so you're going to tell your friend who's going to drive, I know the minimum number of miles. He'll say, so what? Where do I go? And you figure out where you go by just turning this on its head. You stand at Toronto and say, oh yeah, I know how many miles, and I can check these three things to see which one of these cities I came from. So then you kind of turn this calculation on its head. Same logic run backwards again. So anyhow, that's the idea. How does this have to do with biology? A lot. One of the wonderful things about protein sequences and nucleic acid sequences is that the sequences have a chemical direction to them. So we do know left from right. Here's this sequence and that sequence. And what do we want to do with them? We're trying to make a matching of letters in, which is the top one. Actually, I didn't get the top one down here, did I? Whatever. Letters in this sequence match to letters in that sequence. It's pretty funny. At any rate, so this A match with A is really going to mean, what we think is that there's an ancestral sequence from which these two sequences evolved, and when we do a matching, we would say these two letters were related throughout history. Those two were related throughout history, and so on. And we're trying to get as many of the letters to line up as possible and to be as like one another as possible. The reason it's hard, one of the reasons it's hard, is that letters can be erased or added. And so the combinatorics gets more tricky. This is something called a global alignment. This is what we were doing with those protein cytochrome C sequences, all 27. Choose two pairs of them. And we want to account in this evolutionary story of all the letters in one sequence with all the letters in the other one. Account for them as being identical or erased or added. And here's another version of something slightly more realistic. It's the beginning of a couple of transfer RNA sequences, and the stars show the steps. And of course, those stars are are cities that we're visiting on this path, and we get out of that an alignment where this time you probably find. So there's a letter that was either added to one sequence or erased from the other, and there's a couple of letters that aren't identical. But you're trying to make these alignments that look as much like one another, and the pairs of letters are cities on our way. At least that's the analogy. And this goes on. Everybody uses Google to find their name, or text searches to find your name on the web. There's a version of this where you have a short sequence, and this is a very popular sequence for promoter sequences of E. coli, T-A-T-A-A-T. And you take this and you're looking for occurrences of that small sequence along this larger one. In this case, here was the best occurrence, and it was a T-C-G-A-A-T. So there were two mispaired ones. And this is the problem that Temple Smith and I worked on, finding two pieces of longer sequences that look a lot alike. And in this case, toward the end of this sequence and the middle of that sequence, we find this line. And versions of this are run probably millions of times each day. This is the sort of the primary use of CPU time on biology databases. BLAST is a version of this, for those of you who know that. So putting together, reading genomes, inferring, figuring out what the sequence of bacterial genome is limited by our ability to only read short pieces at a time. And so maybe it's 700, maybe it's 600, it used to be 500. So you just get to read short pieces. And so what happens is these longer texts are broken up into pieces, and these small pieces get picked at random and read. And so it's just like you had a big newspaper article, you Xeroxed it a whole bunch of times, cut it randomly into small pieces, stirred those pieces up, and then started reaching in and lifting out. And of course, you'd look for text that overlapped one another and say, oh, I think they belong in the same place, and glue them together. So that's just how this goes. Again, the bacterial genomes can be 5 million base pairs long, and you get 600 letter reads. The human genome is 3 billion letters long, you get 600 letter reads. So this is a challenging problem. And the human genome is filled with repeats. There's something called ALU, which are about 320 letters long. We have 1 million of them. 10% of our genome is filled up with versions of that 300 and some letter thing all over the place. Doesn't make it easy to piece the genome together when you get to read 600 letters long. And this is called shotgun sequencing, a very naive schematic that I stole from somewhere. And I love it. This was from a textbook, and I love it when non-statisticians do this. They have random coverage. You can see one marches right along the other, and naturally that doesn't really happen. They get stacked up. But at any rate, you don't know the order they come in, and so you're trying to infer that they overlap by doing sequence comparison with the pieces. The bottom piece goes down on the bottom part here as the arrows go, and then you have to calculate the overlap. And these sequences are 98, 99% correct, but there are errors in them. And so there's, of course, a route finding problem that does that too. And you're trying to detect these overlaps at one end or the other. So you have to tweak these things around, but the same idea can be made to work. And just to give you a hint, if there was a 1.5 million base pair genome and you had 30,000 reads, that would come close to 1 billion comparisons. And obviously, a whole bunch of those can be eliminated. You can speed things up, but there's some serious computation involved here. Think the human genome. So the human genome being sequenced, or a version of it being sequenced, people turn to asking questions about finding disease genes. And so what in our genetic material makes us have propensity to, say, certain kind of heart disease. And so the idea is to take lots and lots of humans, lots and lots of people, assess the differences amongst them, and look for differences that are in common with one of these diseases. That's the simple version, but that's where we're at. Now, there are various kinds of variation between our genome. The most obvious kind are single letter changes, where I have a GC base pair and you have a TA base pair. But there are also insertions, deletions, as I was saying earlier. There are duplicated regions. There are pieces that are in a different order in my genome than yours. So these single letter changes are by no means the whole story, but they're the part of the story we can really access much better. So they even have a special name. These single letter changes are called SNPs, single nucleotide polymorphisms. And our genomes differ on the average of one of these SNPs per 600 to 1,000 base pairs. So there are lots of them. And I love first papers in subjects because A, usually they're easy to understand, and B, they're exciting. So this is the first paper of its sort. And this was from a company called Perligen in the Bay Area. And they took 24 so-called ethnically diverse individuals. We'll come back to race in a little while here. They had some powerful technology to where, our chromosomes comes in pairs, one from our mom and one from our dad, and those can be different. And so they separated out individual chromosomes from these individuals, and they ended up with 20 independent copies of chromosome 21, single, either mom or dad, all the way down. And then they basically resequenced the entire chromosome. And so they found about 36,000 SNPs, single letter changes along this chromosome, which is one of the smaller chromosomes, 30 million base pairs, which is why they chose it first. And what came out of the first looks at this surprised people, and I'll try to explain this slide. So in this slide, the 20 individual chromosomes are columns. So if you counted across there'd be 20 columns. The individual lines are places where there are differences in the genetic material. So maybe on the average, every 1,000 base pairs in the chromosome, there'll be one of those places where some people will have one base pair and somebody else has, at least one other person has a different base pair. And they're just colored blue and yellow because basically there are only two different ones. And so the blanks are just missing data. And you'd imagine that there would be an awful lot of different sequences. So we went up and down here. But people discovered something called haplotype blocks, and we'll show you one here where you draw these lines across here and amplify things a little bit. And what the people in the paper did was they sorted this interval, and they found mainly 4 groups: yellow, yellow, blue, blue, yellow, yellow, etc. And everybody in this first column had the same genetic material for whatever that is, 40, 50,000 base pairs. Amazing. To me, at least, amazing. And there were 4 others and then some minor ones. And this turned out not to be atypical. And there are arguments for why. So when does the mixing come in? The mixing comes in when you pass your genetic material on to your child. And what you'll pass for chromosome 21, some of it might be from your mom, and then there's a crossing over event, and some of it's from your dad. And so that's where really the stirring up of these sequences go. And there are a couple reasons for there being these commonalities or arguments. One of them is just that the human population went through a bottleneck and we haven't had much time to stir it. The other one is that maybe there's some restrictions for where these switches can take place. I don't care personally. I mean, scientifically it's interesting, but emotionally, I'm very taken by this because it says that with Aboriginals in Australia, with Maoris in New Zealand, with Han Chinese, with my French ancestors, there's a huge amount of genetic material we're carrying around that is exactly the same. So anyway, extremely neat. I'll come back to this in just a little bit then. Hopefully I'll set up for my talking about race. So there are lots of computational challenges. Dynamic programming solved some of these as went along, and incidentally was very useful in finding these haplotype blocks when you didn't know them. So you could use dynamic programming for that. But there are all kinds of problems where it's basically of no use. Taking an amino acid sequence and trying to infer the three-dimensional structure of proteins has been a huge problem for at least 40 years. It will be for a long time yet, I'm afraid. There's microarray data, which comes in arrays and assesses the abundance of transcripts from many, many different genes, thousands of different genes in an organism, and analyzing that data, dynamic programming doesn't help. There are genetic networks, how our genes interact with one another. So there are genetic networks. Dynamic programming doesn't help much there. And the hope to use this genetic variation and learn things about genes and environment and human diseases is not a dynamic programming problem either. But it was useful it remains useful for lots of things. So I'd like to use that, our commonalities, our differences, as a boost to start talking about race. And we all think we know what we mean when we say race. But I don't think very many of us actually do know what we mean when we say it. So there's a bunch of definitions. Unscientific term for a group of related people sharing physical characteristics. That's not a very precise definition, but it's a definition. Distinct genetic group characterized by traits transmitted to offspring. So I guess if I can see, if I have a long nose and my kids have a long nose, there you go. Each of major divisions of mankind, and that's done somehow by physical characteristics, I have no idea. The one that I personally am most at ease with, and what I think most of us mean, it's a group that was more or less isolated geographically, culturally perhaps, who share a common gene pool, who have genetic variations that they share among themselves, which tends not to be in the other races. And I'm a trout fisherman. And if you look at a map of the western United States, there are river drainages where certain subspecies of trout live, and they have names, and I can tell them. So in some sense, I fit in the last one. Just to challenge you with how interesting this subject is, here's a picture of 12 school children from New York City, just from the common school system in New York City. Not only do I think they're really interesting to look at. You probably can't read it. I hope I can here. I'll just start, and I'll read across the top. The kids' names are there. I won't read them. I'll read their stated race, national origin. So the first girl is Pakistan African American. Second girl, Finnish African American. Third, Japanese African American. Fourth, Russian, Polish, Jewish, Puerto Rican. Let me get the second. So I'm starting off with Lisa here, the girl right there in the second row. Colombian, Scottish, Irish. Hawaiian, Chinese, Japanese, the second girl. Dominican, Russian, Jewish. He doesn't look happy, does he? African American, Pennsylvania, Dutch. Amazing. Okay. So race is, of course, a topic Americans worry about a lot. And to kind of carry along just to what I've just been saying, most of us definitely, we see someone, we say, he's African American, he's, whether Chinese or Japanese, I can't tell, but certainly Asian. And what are we responding to? I think mostly we're responding to skin color, but sometimes it's height, and you know, that stuff just goes away, right? You feed a nation that tends to be short, and you feed them a lot, and they get tall. So the height stuff just isn't going to work. Size of nose, that's a popular one. And there's this wonderful scholar, Kenneth Prewitt, who is a social scientist, and when he was writing an article about political issues of race, he said, there's no science to turn to. Well, until recently, there wasn't a lot of science to turn to, but now there's just a little bit of science to turn to, and it's really interesting, to me at least, to take a look at it. A lot more is coming. In the 1980s, you couldn't get a grant that was connected to studying race in the US. It was just not. But after the Human Genome Project got there, you couldn't avoid facing up to this. But I'd like to give you, again, coming from the US, but I think this is indicative of people's attempts to grapple with this. We have a census. We count people every 10 years. We begin that process in 1790. And immediately, the issue of race in America pop up, or what we mean by race. So there were 4 categories. You were free, which meant you were white, or you were slave. And what is this taxed or untaxed thing? That's to pick out the Native Americans. Native Americans didn't have to pay taxes. And this was done this way, the free or slave. Instead of just counting the whites and forgetting the rest of it, the southern states made this big political push, and they got the slaves counted so they would have more political power. The slaves didn't vote, but they wanted them counted. So, and this stuck with America until approximately 1970. In 1820, there were enough former slaves who were freed that there was a category called free colored people. And this category lasted in the American census until 1970. The Civil War was in the 1860s, to put that in perspective. Chinese got counted in 1870, Japanese 1890, Filipinos, Koreans, and Hindis, which apparently are all one thing, got added in 1920. Once they tried, Mexican people from Mexico, which could mean lots of things, there was a Mexican category introduced, and the Mexican government got really mad, so they took it away. And so that stayed white until 1970, when Hispanic origin came in. So the other races are multiplied by, whatever races we count is multiplied by 2 because you have a Hispanic origin or not. I don't claim this makes sense, I'm just claiming that's what it is. So in 1977, there were 4 races: Asian or Pacific Islander, American Indian or Alaskan Native, black, white, and in 1990, a senator from Hawaii got Hawaiian added in. So there were 5 races multiplied by Hispanic, non-Hispanic origin. That gets 10, but they allow various categories, so that today, at the last census, 2000, there were 126 categories were possible. I don't know what all this means, but it's what people believe. And I just want to go back, and that sounds silly, right? But it's not about biology. It's about culture as well, and politics. And you know, you look at these kids. I'll start off with the little boy with the eyes and the crew cut there. Salvadoran, Guatemalan. Next girl is Hungarian, Japanese. Then the girl following, French Canadian, Jamaican, Irish, Italian. Marvelous. Dutch, Jamaican, Irish, African American, Russian, Jewish. It's the last guy. And that probably means a lot to that kid. It's his family tree. So it's important in that sense. But when people are kept from voting or rights are withheld by which of these boxes you happen to fall into, it's an issue. So just to try to portray for you what people, how people respond in strange ways to things to do with race. You may have heard that there was a heart disease medication that was supposed to be effective in African Americans but not effective in whites. And somebody wrote a paper in 2004 in Nature Genetics looking at that and other medicines. And their article said, this is a direct quote from the paper, the underlining isn't direct, 29 medicines or combinations of medicine have been claimed in peer-reviewed or scientific medical journals to have differences in either safety or more commonly efficacy among racial or ethnic groups. But these claims are universally controversial, and there is no consensus on how important race or ethnicity is in determining drug response. Clear enough. We don't really know, is what they said. The Los Angeles Times. A report in the journal Nature Genetics last month said that listed 29 drugs that are known to have different efficacies in 2 races. So they got it to black and white, and this doesn't stop there. The Times of London. Only last week, Nature Genetics revealed research from University College London showing that 29 medicines have safety or efficacy profiles that vary between ethnic or racial groups. New York Times, our great paper. By one count, some 29 medicines show evidence of being safer or more effective, ... And USA Today really goes off the cliff. At least 29 drugs are known to work differently in blacks than in whites. According to a recent report in Nature Genetics, blood pressure medicines that are standard for whites have decreased responsiveness in blacks. The FDA requires that information be included in drug label. Wow. Now, I couldn't find a quote in the Straits Times, but so let's think about this a little more and show you a little bit of the evidence before I stop here. So the difference between us in this way of counting one base pair over 600,000 base pairs. So 1 letter in 1,000 makes me distinct from you. 1 letter in 100 makes us distinct from chimpanzees. It's closer than you think. Now, so you probably know this out of Africa story. Modern humans have been around more or less 200,000 years. 100,000 years ago, people migrated from Africa, and part of the justification for that is looking at human populations in Africa, which have a lot more genetic variation than the populations that migrated into Europe and Asia and America. So, great. So a really interesting study by Noah Rosenberg and colleagues, they took 377 places on the genome where that were known to be highly variable, but not to have any known phenotypic effect. They got these measurements made with over 1,000 individuals from 52 different populations. The within population differences among these individuals counted for 93 to 95% of the genetic variation. The differences among the major groups, and I'll talk about that in a minute, is 3 to 5%. So, wow, this really important thing called race. But in fact, with this, they were able to find 6 main genetic clusters, 5 of which had big geographic regions associated with it, just what that original definition or the trout version of this would have. And the subclusters often corresponded to these individual groups they'd sampled. Here's a little look at their data. I'll read so each line goes with one of these markers, and this is basically allele frequency, and a different color is just a different allele. So, and I'll read the countries: Africa, Europe, Middle Eastern, Central South Asia, East Asia, Oceania, and America. And so we go across the top, well, either one. Maybe I'll do this one's easier to reach. So there's a little difference here, but not a huge difference even between Africa and Europe with that one. So you go across, I don't know which ones would have big. Oceania, this is quite different, but probably a smaller population going in there. So they really were able to find out what looks like major geographic groups, major racial groups, using this data. But again, think about most of the variation were within the populations, not between them. So not a huge number of differences that made it. So I'm back to this skin color thing, because I think that's what most of us see. Dark people, light people. And zebrafish was pioneered here as a model organism here in Singapore. I'll have the date on this. It's not too long ago that a mutant zebrafish was discovered that had a different color. And as you probably know, melanin is this pigment that gives our skin the color. And what this is the normal fish is on top, and it's showing the melanosomes, the clumps of pigment there. On the top, it's quite dark, and down here, this poor guy is, has law. He doesn't have it. So what's going on there? And they found a single gene that caused this difference. This is December 2005, and that gene has a homolog in us. We carry that gene, and in fact, one mutation in that gene accounts for 25 to 38% of skin color differences between Europeans and sub-Saharan Africans. One click, one change. And we've got this huge component of skin color. Now, the skin color, a couple of things about it. No one quite knows how many genes are really involved in skin color, and maybe hundreds. It's certainly a bunch. People pick off the top 5 or 8 or something. This is clearly one of them. And there's a lot of controversy about whether skin color was selected for. Jared Diamond, who writes those wonderful guy at UCLA who writes very books, I think Catastrophe and Guns, Germs, and Steel. He thinks that the difference in skin color is entirely sexual selection, is that people choosing one another because, dark is sexier or dark is not sexier. The other explanation was that the Norwegians who got from Africa up there in that place that the sun doesn't come out very much. If you've ever been in Norway, when the sun finally comes out, people are right out there in the sun trying to soak some up, that to get vitamin D, they needed to cut down on the interference. None of these is completely known or settled, but it seemed to me that well, the other thing is that these changes had to have taken place in 100,000 years. So it couldn't be incredibly sort of deep in some network. It couldn't be incredibly complicated, because there just hasn't been, there have been generations, there are differences, we love the difference, we love our family trees, some of us do get sick or tendency to get sick for genetic reasons. These things count, but this race business is not very important, and I wish the world knew that. And I'd just sort of like to leave my final lecture, I'd like to leave these lovely fish up there with their racial differences. Thank you very much for listening. Yeah, if there are any questions, it will be easy to ask me questions I don't know the answer to, but feel free. Well, people have written probably hundreds of papers now about this. And one of the ways, originally, you looked for, if you went back to just finding genes in E. coli, which is simpler, you would look for, codons that went together until there was a stop codon, and that would be unusually long. People look at the statistics of the letters in the codons if they're different distributions, so there's a statistical aspect to this. But we don't do it perfectly. By no means, yeah, by no means do we do it perfectly.
So when we say that we have a 100% quality and very good quality sequence in databases, that still is not.
First of all, I wouldn't believe the 100% quality, first of all, because these are all experimental processes, so they're almost sure, there are mistakes in all this stuff. So you can't believe it's perfect. And even if it were 100% letter correct, finding all the genes is a hugely challenging problem. Sure.
Just was curious, you mentioned if I have a gene or protein coding gene, if I change one letter, a lot of variation comes in. There is variation in terms of phenotype, even if a one nucleotide.
Yeah, well, the thing I just showed you, that's the result of A to a C, I think, in the coding region. And what it ends up doing is basically it makes the protein shorter for some reason.
So my question is, from statistics point of view, if I am extracting features from the sequences, the full sequence if I use, and if one letter changes, my picture wouldn't change in the numbers much. Then
That's right, and so you realize you've got the problem of you're trying to find the gene that influences some disease, and you've got billions of letters genome, you've got variation, you've got the natural variations, you've got sequencing errors. It's a really hard problem. Even the one gene, one disease kind of thing works pretty well, but several genes, these weaker signals, because they all contribute a little bit, really makes this genetic disease